Is Jean Marie P. Hapitan, your reporter for today. My report is all about animal development, stage of development, the fertilization. Animal development, the process that led to the formation of a new animal, is starting from cells derived from one or more parent individuals. Development occurs following the process by which a new generation of organisms is produced by the parent generation. The purpose and stages of development. Your body consists of over 30 trillion cells, but you began as a single cell, a fertilized egg or zygote. As an animal, embryo develops, its cells divide, grow, and migrate in specific patterns to make a more and more elaborate body. Plant cells perform differential expansion instead of migration. To function correctly, that body needs well-defined axes, such as head versus tail. It also needs a specific collection of many-celled organs and other structures, positioned in the right spots along the axis and connected up with one another in the right ways. Stages of development, the fertilization. Development Step 1. Fertilization is the process in which a single haploid sperm fuses with a single haploid egg to form a zygote. The sperm and egg cells each possess specific features that make this process possible. The egg is the largest cell produced in most animal species. A human egg cell is approximately 16 times larger than a human sperm cell. The egg of different species contain varying amount of yolk, nutrients to support growth of the developing embryo. The egg is surrounded by a jelly layer composed of glycoproteins, proteins that have sugar stuck to them, that releases species-specific chemoattractants, chemical attractors, that guide sperm to the egg. In mammals, this layer called zona pellucida. In placental mammals, a layer of follicular cells surrounds the zona pellucida. The zona pellucida jelly layer is separated from the egg by a membrane. Just underneath the egg's plasma membrane are cortical granules, vesicles containing enzymes that will degrade the protein that hold the vitelline envelope around the plasma membrane when fertilization occurs. This diagram shows that this color sky blue is the haploid nucleus contains one copy of each chromosome, the cytoplasm, and this color violet is the mitochondrion, produces ATP used for energy, contains mitochondrial DNA, which is passed to offspring. Cortical granule. This color green is the cortical granule. Releases enzymes during fertilization that harden the zona pellucida and digest binding proteins on sperm to prevent polyspermy after a sperm cell has entered the ovum. And this color orange is the first polar body. And the zona pellucida jelly coat contains glycoproteins that protect the inner contents of the ovum until the acrosome reaction of fertilization helps prevent polyspermy. Nucleus of follicular cell. And the last one is the corona radiata. Follicular cells protect the inner layers of the ovum and helps prevent polyspermy. Sperm. Sperm is one of the smallest cells produced in most animal species. The sperm consists of head containing tightly packed DNA, a flagellar tail of swimming, and many mitochondria to provide power for movement. The plasma membrane of the sperm contains protein called bindin, which are specific proteins that recognize and bind to receptors on the egg plasma membrane. In addition to the nucleus, the sperm head also contains an organelle called the acrosome, which contains digestive enzymes that will degrade the jelly layer, zona pellucida, to allow the sperm to reach the egg plasma membrane. This is the part of the sperm. The head contains of acrosome, plasma membrane, and nucleus. The midpiece contains of centriole, mitochondria, and flagellum. and the end piece contains of eggshell filament and tail. Fertilization of sea urchin. Sea urchin are spiny globular echinoderms in the class Echinodea. About 950 species of sea urchin live on the seabed of every ocean and inhabit every deep zone from the intertidal seashore down to 5,000 meters. The spherical hard shell of sea urchin are round and spiny, ranging in diameter from 3 to 10 centimeter. Four important events of fertilization in the sea urchin: the acrosome reaction of the sperm-egg fusion; third, the cortical reaction; and the last is the formation of the fertilization cone. Male and female sea urchins are easily induced to shed massive numbers of egg or sperm. The eggs are mature at release and are fertilized externally simply by mixing eggs and sperm. The resulting embryos are optically transparent and develop synchronously in nothing more than in seawater. Step, Steps of fertilization in sea urchin. First is the sperm is attracted to and contacts the jelly layer zona pellucida. Second, the interaction between receptor on the sperm cell and glycoproteins on the egg cell initiates the acrosome reaction. Digestive enzymes are released from the acrosome and they destroy the jelly layer, a zona pellucida, thus creating a pathway for the sperm to approach the egg. Third, the sperm reaches the egg plasma membrane and the sperm and egg plasma membranes fuse. This reaction is mediated by the sperm binding proteins, which bind to binding receptors proteins on the egg plasma membrane. This interaction is specific. This interaction is species specific with slightly different versions of the binding protein and the binding receptor proteins in every different species. Fourth, the fusion of the sperm and egg membranes initiates electrical depolarization of the entire egg plasma membrane temporarily for 10 to 20 seconds, preventing any other sperm from fusing with the egg plasma membrane. This membrane depolarization, mediated by an influx of sodium ions, is the fast black polyspermy. Fifth, the membrane depolarization initiates across the wave of calcium release across the plasma membrane. Next, the calcium wave initiates the cortical reaction in the egg. The cortical granules fuse with the egg plasma membrane, releasing digestive enzymes that degrade the binding receptor proteins that serve as sperm docking site and the proteins that hold the vitelline layer against the plasma membrane. 7. The cortical reaction result in lifting away of the vitelline layer away from the egg plasma membrane to create the fertilization envelope. The fertilization envelope is a barrier that prevents additional sperm from reaching the egg and is the slow block to polyspermy. And the last is these events culminate in egg activation, causing the egg to recognize that fertilization has occurred and resulting in initiation of development. This diagram shows the fertilization of the sea urchin. Two types of fertilization: internal fertilization and external fertilization. Internal fertilization, most land, both invertebrate and vertebrate, use internal fertilization. In mammals, reptiles, and birds, the gamete meet inside of the female's body. This is called internal fertilization. All land dwellers need to fertilize this way because sperm prefer for swimming to walking. Internal fertilization protects the fertilized egg or embryo from predation and harsh environment, which result in higher survival. Internal fertilization is characterized by sperm fertilizing the egg within the female. The three methods include the oviparity, the egg laid outside female body. Ovoviparity is the egg held within female, and the viviparity is the development within female followed by live birth. Example of internal fertilization. The fusion of the gametes occur inside the female body and the zygote fertilized egg is formed inside the female body. External fertilization is a strategy of fertilization in which a sperm cell unites within an egg cell in the open, rather than inside a specialized organ within the bodies of the parents. External fertilization is limited essentially to animals living in an aquatic environments. The flagellated sperm must have fluid in which to swim, and the eggs lack a protective coat or shell so that sperm can penetrate and fertilize them and would dry out in the air. Almost all aquatic invertebrates, most fish, and many amphibians use external fertilization. Example of external fertilization is fish. We can see that male fish waits to spread the sperm over the eggs, and the female fish lays egg. External fertilization occurs outside of female parent. Example is fish, amphibian, and aquatic vertebrates. And that's all. Thank you.